Hey there. My name is Sherwood, and over the summer I did research in Professor Burk's lab where I studied orexins and the decline of attention with age and its correlation to Alzheimer's. So, for a little background information about all of this, Alzheimer's is this age-related brain disorder that is commonly shown by this cognitive degeneration, which is shown in the symptoms such as memory loss, confusion, difficulty learning new things, and our focus of a loss in attentional processing. Now, in order to study this attentional or loss in attentional processing, we looked at these things called orexin neurons, which are these brain cells that are located in the hypothalamus of the brain, and they release these neuropeptides called orexin A and orexin B. These peptides essentially just stimulate this other brain system called the basal forebrain cholinergic system, or BFCS, which helps regulate these really important processes such as reward, appetite, learning, and cognition. As you can see in the image provided, the orexin system, which is circled in green, will interact with the BFCS circled in yellow. And then from there, that in turn will then regulate these things such as reward, appetite, arousal, movement, and then cognition, which includes our focus of attention. So this kind of information then spurred our questions of if we see or cause a loss of erection projection in the brain, does that in turn reduce attention over time? And then if we do see that loss in attention over time, how might we then apply that to the brain disorder of Alzheimer's? Now, to start off with our study, we used 24 rats, and we had 12 rats be lesioned by receiving a neurotoxin called orexin B saccharin, which is essentially just depletes the orexins in the brain. And the other half was just a control group that received a vehicle injection that didn't produce any orexin activity. And then after fully recovering, both of these groups were then trained in a two-lever sustained attention task, which I'll explain here. So this is the two-lever attention task. And it's actually, the components of it are quite simple. We have a light that will turn on and off at different intervals. We have two separate levers, and then we have a dispenser for water. Now, essentially how it works is that the light will either turn on or not turn on at particular intervals. And if it does turn on in an interval of time, the rat will say, oh, I've been trained to know that now I have to push the left lever if I want a reward. And if it successfully does that, then it will receive the reward of water. On the other side of things, if the light doesn't turn on in an interval of time, the rat will know, oh, now I have to press the right lever or the other lever if I want to receive a reward. And if it does that correctly, then I'll get water. And this task can be done at multiple different difficulties where either the light will be on for longer or for shorter periods of time, or the intervals of time between when the light should turn on will either decrease or increase in order to get a substantial measurement of attention. So because unfortunately this is a very age and time intensive study, we only have our hypothesized results currently. So we plan to study these rats over their complete lifespan so that we can see, oh, how does the accuracy of their attention compare as they continue to increase in age, such as how with like Alzheimer's, for example, you don't really see those attentional losses all at one point. It's something that gradually happens with age. And so we anticipate, however, that as we continue this study over the rat's lifespan, the attentional accuracy of rats in the lesion group will slowly decline, while the accuracy of the control group will remain constant. So although we don't really have our completed data recorded yet, if our data or future data that we expect is supported, this would reveal a significant correlation between the degradation of orexin and the decline of attentional processing that is present in Alzheimer's. So these results would be really beneficial not only for like learning more about what might influence this loss of attention, but it can also bring in a lot of new studies that are looking at these treatment methods. So, for example, future studies might start research on these test drugs that may re-stimulate orexin activity in the brain. And then by doing so, then reverse some of the cognitive deficits, such as these attentional losses that were caused by a lack of orexins. But yeah, that's what I did over the summer, and I had a lot of fun. And yeah, thank you.